So, good morning, everyone. My name is Hussein, and today we'll be continuing with the first chapter, The Living World, and we'll be seeing the final part of the first chapter, which is nothing but taxonomical aids. Okay, so under this, we will understand the 9 taxonomical aids which are used in the field of taxonomy in order to study basic processes of taxonomy, which were characterization, nomenclature, classification, and identification. Yes. So before we start, let us look at the list of taxonomical aids. So we have 9 taxonomical aids which we'll be studying today. We have herbarium, botanical gardens, zoological parks or zoos, museums. We have key, flora, manuals, monographs, and catalogs. Okay, so going to the first one, which is herbarium. Herbarium is nothing but a storehouse of plants. Okay, of plant specimens. So in this storehouse, the plants are preserved by drying and pressed in dried and pressed form. Okay, and they are arranged according to the universally accepted classification. Okay, so we see in the picture on the left picture that how a herbarium sheet looks like and how a plant specimen is stuck on the herbarium sheet. Okay, so we'll also see how the labeling of the plant is done. Okay, so herbarium sheets are labeled with information about the date and place of collection, English, local, and botanical names, the family to which the plant belongs to, the family of the plant, and the collector's name, the person who collected the plant and made the herbarium sheet. Okay. So the collector's name and the person who made the herbarium sheet can be different as well. Okay, but the collector's name is what is to be mentioned. Okay, the label is generally situated at the right hand bottom corner. This is a neat question. Okay, the label is generally located or situated at the right hand bottom corner. Okay, yes. So and after making the herbarium sheet, it is pressed and then stored as given in the picture in the right hand side. Okay. So the standard size of the herbarium sheet is 11.5 inches into 16.5 inches. This is breadth by length. Okay, and in centimeters it is 29 by 41.5 centimeters. Okay, so during the preparation of this herbarium sheet, the plant sample is sprayed with mercury chloride, which acts as a pesticide. Okay, so obviously a plant is a living organism, so once it is plugged and it is placed on the sheet, microorganisms or be it bacteria or fungi might attack the plant. Okay, and try to eat it off. So to avoid the degradation of the plant, we spray it with mercury chloride, which acts as a pesticide and kills all the insects, bacteria or fungi which might infect the plant. Okay, thereby preserving the plant. Yes. So care is to be taken that the plant specimen chosen is not damaged or diseased. So yeah, so when choosing a plant to prepare a herbarium sheet, care has to be taken that the leaves do not have any holes in them, or they do not have any notches in them, or the flowers are not infected, and that all the petals, all the floral parts in the flower is present. Okay, so all these care should be taken. And on the right hand side, you see that the label is given. So it is just an example of a label. Okay, how a label should be. Okay, so what are the uses of herbarium? Herbarium is used for quick referential studies. Okay, and alpha taxonomic studies, which is taxonomic studies which focus on the morphological characters only to study a particular plant. Yes. Coming to the next taxonomical aid, which is botanical gardens. Okay, so these are specialized gardens having collection of live plants. Okay, living plants for reference and identification. Each plant is labeled with its botanical name and family. Okay, so some of the famous botanical gardens pictures are given. On the top, we see that it is National Botanical Research Institute, Lucknow. Okay, that is the top picture. In the bottom left, we see the Kew Herbarium or the Kew Botanical Garden, which is also the largest botanical garden in the world. Okay, and then on the right, we see the Indian Botanical Garden in Howrah. Okay. Next, the next key is the next taxonomical aid is biological museum. Okay, which are museums. Okay, so it is nothing but the collection of preserved plants and animals for study and reference. Okay, so there are plants stored as herbarium and as what do you call it, preserved dead what do you call it, specimens. Yes, and animals are stored in various forms. Okay, So in a museum, the specimens preserved in preservative solution in jars and containers are seen, as seen in the picture. We have dried specimens of plants and animals also seen. Okay, then we have insects preserved in the form of insect boxes, as seen in the picture again, and stuffed larger animals like birds and mammals are also seen. Okay, and collection of animal skeletons, animals which are extinct. Okay, whose bones or skeletons are only present, are also what do you call it, present in the museum. Okay, and information about them is also present. Yes. So the next taxonomical aid is zoological parks, or as we call it in layman's term, zoos. Okay. So these are places where live wild animals are kept in protected environments under human care. So zoos give us an insight into the food and behavioral habits of the animals. So why do we keep the animals in zoos? To study the food habits and the behavioral habits of the animals. Okay. So yeah, a place where birds are kept is called an aviary. Okay. The next aid is key. So what is a key? It is nothing but an analytical method for identification of organisms. Okay, based on similarities or dissimilarities. Okay, so it is based on the concept of contrasting characters, generally in a pair. So how we use key is that we have a pair of contrasting characters, and we see whether the organism goes or has one of the characters or not. Okay, so this pair of contrasting character is called a couplet. So this pair of contrasting character is called a couplet. When we go into the example, you will get a clearer idea. Okay, so each couplet has two opposite options. Okay, contrasting character. Of these, only the relevant option is accepted, the other is rejected. Only the relevant option is accepted, the other one is obviously rejected. So it is a yes or a no answer, a positive or a negative answer, a black or a white answer. There is no in between. Okay, so each statement in the key is called a lead. So the pair of contrasting characters is called a couplet, and each statement is called a key. Okay, the ultimate lead. Sorry, the ultimate goal of a key is the identification of taxa. It may be at the genus level or at the species level. Okay, so there are two types of keys: indented keys and bracketed keys. Okay. So examples of key representations, let us look into the first one, which is the branched tree key. Okay, where a pictorial representation of the what do you call it, contrasting characters is given. So first we start with the invertebrate phyla, which is in which we have two contrasting characters. It is either symmetrical or asymmetrical. So when we know that a particular phyla is asymmetrical, there's only one phyla which is asymmetrical, which is porifera. Okay, so we separate it out. And under symmetrical, again we see that there are two branches. Okay, so one is radial symmetry, the other one is bilateral symmetry. Again, radial symmetry comes out or brings out one separate phyla, which is Cnidaria or coelentrates. Okay, followed by which in under bilateral symmetry you see whether anus is present or not present, meaning whether the digestive system is complete or incomplete. So we see that the Platyhelminthes, phylum Platyhelminthes does not have any anus. Okay, whereas all the other phylums have anus. So under anus also, again we divide it into two parts, whether visible segmentation is seen or no visible segmentation is seen. We see that phylum Mollusca does not exhibit any visible segmentation, whereas three other phyla exhibit segmentation. And then under what do you call it, segmentation, under segmentation we see that either exoskeleton is present or exoskeleton is absent. So exoskeleton is absent in Annelida and present in Arthropoda. Okay, so this is how, with the help of contrasting characters, with the two contrasting characters, you what do you call it, separate out individual, it can be species or individual phyla or individual animals or whatever you're trying to identify. Okay, so this is one type of key. The second type of key is dichotomous key, or under dichotomous key there is a type called linked key. Okay, so linked key is a type of what do you call it, bracketed key. Okay, so here what happens is you see that the pair of contrasting characters is numbered as 1a and 1b. It either has fur or does not have fur. If it has fur, then it belongs to Mammalia. If it does not have fur, then they ask us to refer the next pair of contrasting characters, which is it cannot change location or move on its own. It can change location or move on its own. Okay, so For number 2A, they ask us to go to number 3, which gives us the, so once we know that it cannot change its position, or it cannot move on its own, we know that it's a plant. So based on plant's characters, the third one is having two contrasting characters again. Okay. Similarly, the fourth one is an animal contrasting character. So this is how, using what do you call it, consecutive contrasting characters, we single out different groups of organisms. Okay. And the last one is nested key, which is similar to branch key, but it is in a horizontal tree formation. Okay. So every arm of the nested key gives you one organism. Okay. That is the thing. Yeah. So the next key is, or the next aid is Flora. Okay. So Flora and the following aids are all books. Okay. So in order to avoid confusion, let me explain it to you in a layman's term. Okay. So first one is Flora. So Flora gives you an information about organisms in a particular region. Okay. So for example, let us look at the example. We have Flora of Central India, algal flora of Kerala, or moss flora of Rajasthan. So it is telling about a particular region and what kind of organisms are present in the region and where it is present. So not only the habitat but also the distribution of the plant is for a particular area is what is given in flora. है ना? Next one is manuals. Manuals is nothing but the information for identification of names, okay, of species. Meaning that you see that a particular plant you want, okay. So for that plant, what kind of information, how to grow the plant, or is the plant does the plant give us any useful items or not, and what kind of molecules are present in the plant, what kind of molecules can be derived from the plant. So all those kind of information is given in manual, okay. And how to identify the plant, okay. So all those information in a particular area, okay, for a particular area again. So as you see in the examples, we have manual of cultivated plants, we have manual for identification of medical bacteria, we have a manual of medicinal and aromatic plants, meaning that these books help us to identify what kind of plants are there, okay, and their name, and what is there in the plant and stuff like that, okay. So it is helpful in the identification process, okay. Monograph. Monograph are books or records which contain information for one taxon, only one taxon. It can be only for a family, or it can be only for a order, one particular order. For example, you see the example, the Gingiberaceae crops. Okay, so it is only talking about the Gingiberaceae family. Okay, the ginger family, and whether its usefulness, and what kind of medicine has been obtained, and what kind of recipes, or what kind of any information regarding that family is given in the book. Okay, then second example, we have sulfur bacteria. Okay, just on sulfur bacteria, there is a book written. So such a book which explains only one taxon is called a monograph. Okay, it gives all kinds of information about the particular taxon. Okay, and the last but not the least is the catalogs. Okay, catalogs are nothing but alphabetical list of species. Okay, for a particular group, let's say in example, the leftmost example is marine mollusks. Molluscans present in the sea and oceans. What are all the molluscans present? The list of all of them, along with their pictures and other information, basic information, is given in catalog. But the main point is that it gives an alphabetical list of species in a particular group. It can be mollusks, it can be flowers like Indian orchids, or it can be Indian insects. Okay, any group. So again, let me revise. Flora gives you the habitat and distribution of the plant in a given area. That is the important point. Manual gives us the information for identification of a plant in any area. Okay, in any area. Okay. Monograph gives you the information about only one taxon, and catalog gives you an alphabetical list of species of any particular group. Okay. It gives a list of all the species present in the group. Okay. So with this, we come to the end of chapter 1, the living world. Okay, with this I want to end with a, what do you call it, a quote from Carolus Linnaeus. He says, if you do not know the name of things, the knowledge of them is lost. So always know what you're studying about. Okay, always know the thing, or be a little bit curious about knowing the names of organisms, and learning about them and stuff like that. So this is me signing off, Hussein. Thank you and have a nice day.